Students, today we will study sexual reproduction in Volvox. Sexual reproduction in Volvox usually takes place toward the end of the growing season. It is of oogamous type. The species of Volvox are either monoecious in case of Volvox globata or dioecious in case of Volvox aureus. Sometimes colonies of Volvox aureus have both the antheridia and oogonia. The monoecious species are protandous, that is, antheridia mature before oogonium. Usually, a colony involves sexual reproduction, does not have asexual daughter colonies. The cell developing into reproductive structures are located in the posterior part of the colony, and these cells are called gametangia. Usually the gametangia are limited in number. The male and the male and the female gametangia are called the antheridia and the oogonia respectively. Antheridia, the gametangia developing into antheridia, is larger than the vegetative cell of the colony. It withdraws its flagella but remain connected with one another vegetative cell by cytoplasmic thread. The protoplasm of the gametangium divide repeatedly to form 16 to 512 rod-shaped cells. Each cell develop into a naked, biflagellated, fusiform male reproductive cell or antherozoids. In some species, all the antherozoids in an antheridium form a bundle and are also released in a bundle form. In some species, usually those in which the protoplast of the antheridium divides to form more than 128 antherozoids, the antherozoids are arranged in a hollow sphere which shows inversion like the asexual daughter colonies, and the antherozoids are released individually. Next is the oogonium. The gametangium developing into oogonium is also larger than other vegetative cell in the colony. It increases in size and eventually become flax shaped. The entire protoplast of the oogonium finally forms single uninucleated non-flagellated spherical egg. The egg has a partial chloroplast and as such is green in color. It is laden with reserve food material. The beak of the flax shaped oogonium open at the surface of the colony. Fertilization. The antherozoids after liberation from the colony swims in the medium. If then they come in contact with a vegetative colony, they simply swim over. But when they come in contact with the female colony, they get attached to its surface and break down the colonial matrix of the colony in the area of their attachment. This task is probably accompanied by the proteolytic enzyme secreted by the antherozoids bundle. When the antherozoids are in a bundle, the bundle breaks down and the antherozoids are liberated in the gelatinous matrix of the hollow female colony. One antherozoid fuses with each egg to form the zygote. The zygote secretes a three-layered thick wall which may be smooth as in Volvox globata or spiny. The wall contains hematochrome which impart red color to the zygote. The zygote may remain dormant for quite some time and is then liberated by disintegration of the parent colony. Germination of zygote. The zygote usually does not germinate until the following spring. Before germination, the diploid nucleus undergoes meiotic division, forming four haploid cells. These cells undergoes further division. The outer layer of the zygote plate and the two inner layer protrude in the form of a vesicle. The cell migrate into the vesicle. The species of Volvox considerably develop into new colony. For example, in Volvox aureus, Volvox minor, the protoplasm of the zygote divide repeatedly and form new colony by the sequence of Plancke stage as is found in the asexual reproduction. In Volvox companies, Campaniensis, the protoplasm of the zygote, form many biflagellated zoospore, but only one of these survive and the other degenerate. The surviving zoospore come out of the vesicle and swim freely in the medium. It divides to form about 500 cells which arrange themselves in a hollow space. This hollow space undergoes inversion as asexual daughter colonies and develop into new colonies. Thank you.